Okay.
Oh, no, I just muted myself. There we go.
I think so.
Running?
Well,
Yes.
okay.
Okay, so we're going to get started. So today we're here to hear Dena tell us, give her exit seminar, the UC Davis equivalent of a defense, since we don't actually defend our dissertations. But Dena is finishing up with just a complete draft of her dissertation. And
last Friday.
we're all excited to hear about it. So Dena came to us from Portland State after doing a master's at Portland State University. And remembering back to my own defense, I decided that I was going to dig into Dena's application to Davis to come up with something embarrassing.
Oh my gosh.
She said Dena for her dissertation to show how completely different it turned out, except that, you know what? It didn't work. Because what you said in your application to UC Davis was that you wanted to experimentally investigate the effects of fragmentation on primate behavior, diversity, and ecology, which, as we're about to hear today, is kind of almost exactly what she said is almost
a part of it.
a
Yes.
big part of what she ended up doing. She did, however, say that, which made me chuckle. As Dena will tell you, she worked at a site called SAFE, which is a forest fragmentation experiment. In her proposal to come to UC Davis, she said that she was in fact going to fragment the forest herself in conjunction with logging companies.
I was actually really upset when I found out they'd already done it. Like.
Anyway, Dena has had a really and has had a great career here at UC Davis. She's been very successful. She's been awarded 2 Fulbright fellowships, one to do the work she's going to tell you about today, and more recently, a second one to go back to Malaysia and continue this research in exciting new directions. She's had an amazing suite of publications, both in the last few months, but then also a whole bunch coming out, including in a number of very well-respected journals, Journal of Bioacoustics, International Journal of Primatology, and one that I'm quite excited about that's going to Proc B hopefully soon. Anyway, as you'll hear, as Dena will tell us today, she's working on gibbon vocalizations, both the ecology of them, and then also vocalizations as an interesting measure of evolution. And Mark had a few things that he wanted to say about Dena, but before I get there, I just wanted to say, after the talk today, we're going to have champagne, fruit, cheese, food at my house, which is 207 Cottage Circle. It's about a 5-minute walk from here, if you walk slow. So please come and celebrate this wonderful accomplishment of Dena's.
Thank you.
I'm going to be quick because I want to give Dena plenty of time. I've been working with Dena for a year and a half, and there are things that you might not know about Dena. One of them is that she is amazingly good at computing. I mean, you come across as kind of bubbly and
not as much of a computer nerd as I might be.
Not as much, but she is she's one of the true computer nerds of the program. The thing about Dena is that she's not afraid of breaking stuff. and it works for her. So we would talk. I never really told her how to do stuff. I would just say, Dena, somehow we need to figure out how to do this, this, and this. She goes off and she just figures it out. And so she's really good and a good model for all of you who come in who may not have real deep computing backgrounds, but you can just do it and to not be afraid of breaking stuff and fixing it later.
The bull in a china shop with coding.
Can somebody in the background turn down half the lights, not the whole way, but one of the two? Otherwise it gets absolutely pitch black in here.
Gotta get the mood lighting. There we go. All right. Well, thank you all for being here, and thank you guys both for the wonderful introduction. And so I'm going to do my best to cram 6 years into 45 minutes. We'll see how that goes. And so just briefly, as Meg mentioned already, my dissertation research focuses on measuring, quantifying, and understanding processes of primate vocal variation. And I also, in various ways, attempted to shine light on the evolutionary mechanisms that lead to that variation. And I realized over the course of my PhD that that's something that is more of a career-long goal. So I'll talk about the different patterns of variation I found and kind of the evolutionary mechanisms that I think may have led to that, but there's still a lot more that needs to be done. And I apologize, the text is really small, and this is really not a comprehensive list, but they say that it takes a village to raise a child. It definitely takes a village to raise a PhD student, and so I want to say, first and foremost, thank you to Meg Crowfoot and Andy Marshall, who's actually attending here via Skype from the University of Michigan. So to both of you for really helping me through this process, and then, of course, Mark Grote for his help, his statistical advice, counseling, and not just counseling about statistics, about life in general, and how to make it through your dissertation. He was just a wonderful support towards the end. And then, of course, everyone else in Malaysia and here that helped me get through this. And I would go through the whole thing, but I wouldn't have time to actually talk about my research. So, okay, so thinking about how to present the results of my dissertation in a comprehensive and a cohesive manner. I'm going to come back to this slide quite a few times throughout the course of my talk, and I realized as I was making this slide, like, this pretty much sums up my research. So, you know, I can just tell you about this slide and we can go have a drink. No, but obviously there's a little bit more to it. But what I want you to kind of think about and how I'm going to organize my chapters is thinking about vocalizations and acoustic data in general, and trying to understand how they vary and the fact that they vary kind of at these different levels. So acoustic data and data in general is in this kind of multi-level or hierarchical organization. And so I'm going to start small. And so for my first chapter, I focused on looking to see if there are trade-offs in the production of a single gibbon call. And so I'll go into detail what these little things are up here. They're called spectrograms. They're a visual representation of the call. And so for my first chapter, I wanted to know, are there trade-offs in what's going on in terms of the note, specifically understanding if there's a trade-off between note repetition rate and note bandwidth. And then thinking about the next level, so starting small, moving out, I'm also going to show you some data and start to think about the question of how variable are calls within a particular or a single individual. And this could be interesting for a variety of reasons. Maybe people would want to understand, do animals alter their vocalizations in regards to the social context that they're in or the emotional state that they're in? There are a lot of different reasons why you would maybe want to know what's going on here. And then the next level out, working to see if calls are distinguishable between individuals. So does individual 1 have calls that sound and that we can detect are different between individual 2? And then there are a lot of interesting questions that you can address at this level. So not just if you can quantify individuality, but working to understand, do others pick up on these individual cues? And through the use of playbacks, there's a lot of evidence that animals can detect differences in individual vocalizations. And then also, as Meg mentioned, there are conservation applications, particularly for this level. If you can identify individuals just based on their vocalizations, then you can track and monitor them over time non-invasively. And then lastly, the last kind of level for my research is thinking about if there are interesting site or population level differences. And so the first step obviously is to go and see to try to quantify if there are differences. And then if there are, there's a whole suite of interesting evolutionary questions about the types of processes that lead to the site level variation. So this is a really broad kind of overview of how we're going to go through my talk. So first, I want to tell you just a little bit about my study subject. So I focused on the Bornean gibbon shown up here. And so I'm sure not everyone in this room is a primatologist, so just a little background about Bornean gibbons. Bornean gibbons are highly territorial, and they're socially monogamous, so they live in male-female pairs along with their offspring. But of course, the more that you study any animal that you think is monogamous, we see that there's a little bit more flexibility in their social systems, but for all purposes, we can think of them as being socially monogamous. And then something that's really important for me as a researcher is most but not all species of gibbons engage in sex-specific and sex-specific and species-specific duets. And so where the males and the females have different relative contributions to the duet, and they'll go back and forth. And for the research that I'm going to be showing you today, I'm going to be focusing on the female contribution to the duet only. And there are a couple different reasons for that. First and foremost, if you look at or you listen to a recording, the female's contribution is this big, long, beautiful thing, and then the male's little part is just this little dinky bit at the end. So we're actually with one of my undergraduate, or I guess recent graduates, Alice and Lau, we're looking at the male vocalizations, but that's not within the scope of my dissertation. And so I'm focusing on the females, but a lot of these questions are relevant to the males as well. Okay, so just briefly about my method. So the fun part is going out and recording the gibbons. And so I use the same methods for all of my chapters. And so I went out with a digital handheld recorder. And I'm learning now that maybe, well, and for my postdoc research, I won't be going out with these handheld recorders because there are autonomous recorders that you can put out that are waterproof that will record 24 hours a day. But I went out there, not necessarily rain or shine because they don't sing in the rain, but you know, rain and early, or sunny and early. And I went out and I recorded them. And it seems when you're out in the forest and you're listening to gibbons like that they're just singing all the time. But when you choose a particular focal group, you'll go and you'll sit in their territory and they don't sing. They'll sing maybe once every 3 or 4 days. And so I realized that I could augment my data collection by doing something called a playback. So I took a big speaker into what I presume to be a territory of a gibbon, and I would call them. And they would come, so they don't run, they brachiate, so they have this kind of motion that they move, and they would come so fast through the trees. And a lot of times if they didn't see me, then they would start to call. And so that's how I was able to augment and get more data. I don't have too much time to go into it. I'll go in a little bit more detail about my first chapter, but there is some evidence that birds will alter some aspects of their vocalizations in response to playback. So that's something that when I'm analyzing my data, I have to kind of think about that. And then data analysis, I used Raven Pro, which is software from the Bioacoustics Research Program at Cornell University. And I created spectrograms, which I'll talk you through a spectrogram in the next slide. And I used the software to identify the gibbon female calls and then also do something called feature extraction, where I estimated note duration, note frequency, different things like that. And I'll go into more detail on that in a bit. And then, as Mark said, so I really like the analysis part now too, which if you would have asked me 2 years ago, I would have said, no way. But all of the analyses I did in R, and it's actually like one of the more fun parts now. Okay. So those of you that maybe are not bioacousticians or haven't studied or worked with me or seen me give a talk before, this is a spectrogram of the Bornean gibbon call. And so this is a visual representation of the sound. On the x-axis, we have time. The y-axis is frequency. And then the color corresponds to the amplitude or the power of the signal at a particular frequency and time. And so assuming that this works, I'm going to try to play one for you now. Yeah. And so this is the female part of the duet. People, when I'm in the field, always think they're birds. They're like, what kind of bird is that? Like, it's not, it's a primate. Okay, so coming back to this kind of multi-level organization that we have. And so I'm going to now work and go into my 3 separate chapters. And so as I mentioned for my first chapter, I focused on trying to understand if there are trade-offs in the production of a single call. And so it's important to note too that these different levels, they're not necessarily mutually exclusive. It's just that I'm emphasizing one level over another or a couple levels over others. And before I get going into the nitty-gritty of this, I want to really thank my collaborator, Russ Cherif, who is not here, but he said that he would be watching on the live YouTube stream. So hi, Russ, if you're there. But if it weren't for him, I would not have had the idea to do this analysis. I met him a couple years ago when I went to the sound analysis workshop at Cornell. And he gave me the idea for the analysis. And he's also kind of helped me go through the weeds in terms of how to measure signal to noise ratio, bandwidth, things that seem like they should be relatively straightforward, but it turns out they're not. And so, again, we have 2 different spectrograms here, and they're from 2 different females. And so I won't play the whole thing for you, but what I want you to notice is that, so we break up, or we as in I, because I'm the first person that studied this particular gibbon species, I break up the gibbon call, the great call, into two separate parts. And so we have the introductory part here, which has the longer intro notes. And then you'll see that there's a shift and the notes start to be repeated at a more rapid rate. And so that point is what I call the trill here. And so for this first part of, or this first chapter, I'm focusing just on the trill. And I'm going to go ahead and I'm going to play just the trills for both, or both of them for you. And you'll notice, hopefully, so this one, it's quite a bit slower. The trill, the rate is quite a bit slower, and then it also has a much higher bandwidth. So bandwidth is the range of frequency. So if you look at the notes here, they're just much wider. And then now I'll play the second one down here. And you can hear it just goes a lot quicker, right? So that is, those are the trills. And so I'm really interested in kind of understanding what's going on when they're doing these trills. And I want to give just briefly a little bit of background. I don't have time to go into the nitty-gritty of these studies. If you'd like to talk about it after, I'm happy to do that. But I just want to give some context. And so one of the first people to look at this trade-off between bandwidth and trill rate was Jeff Potos, and he looked at 34 different species of songbirds, particularly American sparrows, and this was back in 1997. And so he looked for basically just if there was evidence for this trade-off between how fast the notes are repeated and the bandwidth of the notes. And he found in all 34 of these species, or in these birds, that there was indeed a trade-off. And then 20 years later, another study that just came out recently, they were looking at something similar, which I won't go into the nitty-gritty of it, but just know that this is something that in the bird literature, people have been interested in understanding what's going on with this trade-off. But then there's also something that's called the performance hypothesis, or the performance constraints hypothesis. And this posits that it's difficult to produce trills that are fast and that have a high bandwidth, so to do both of those things. And so then, therefore, individuals that are kind of closer to the performance limits of creating these calls may be of higher quality. So there may be a link between how they're producing these calls and the quality of the individual, or maybe the fact that the females think that they're sexier, you know. And so there is evidence that females will react differently to different trills depending on how close or far away they are from their performance limits. And then another really, I think, cool, interesting study was done in neotropical singing mice, and I was really excited when I found this picture. And so they don't actually look like that when they sing, but they do have a trill. And so this paper, I believe, was from 2011. And so they first looked for evidence of this trade-off between bandwidth and trill rate, and they found it. And then they artificially manipulated the androgen, or the sex hormone levels of the male mice. And they found that when they had lower levels of androgens, that they were further away from the performance limit. So there is something that's regulating what's going on in those calls. And then they did playback experiments, and the females preferred to be by the speakers where the calls were more high performance. So there's a lot of stuff going on in the literature right now about what this all means. And so I'm just going to kind of slide in there and be like, all right, well, this is all with males. And now I'm looking at this trade-off in females and in a non-human primate. And so unfortunately, I wasn't able to do any cool kind of playback experiments or alter their hormones, which with the primates, that wouldn't be ethical, or at least gibbons. And so again, this is a spectrogram. I've shown you a couple of these already. It just looks different because I altered the parameters, the settings a little bit. And the reason I did that is I wanted to show you the fundamental unit of analysis here. And so I followed just kind of the convention as to how people study these things. And so what we do is we divide the trills up into 1 second bins. So each of the red squares shows a 1 second bin. And then I had counted the number of notes per 1 second bin. So that was my trill rate. And then I averaged the bandwidth of the notes for a particular bin. And I didn't have time to show you figures from the other papers, but they created something really, really similar to this with the singing mice and the songbirds. And so what I found is that there is a significant negative relationship between trill rate and bandwidth. And so this regression line that I'm showing you here, it was calculated using a 90 percentile quantile regression. And there's a lot of kind of discussion in the literature about the best way to look at this trade-off. And so what this is trying to show is the upper bound as to what's going on in terms of the trade-off between trill rate and bandwidth. And so what we would think, or we could propose, is that females that are closer to this line, these are higher performance calls. And so maybe, and I'll talk about in my future directions, maybe they're of higher quality or something along those lines. And then down here, these are lower performance trills. And I wasn't very happy with this method for a couple different reasons. Well, mostly that I had to average over all of the bins, over all of the calls within a particular female. And so what I'm showing you is each point here represents one female, one gibbon female. And so I was losing a lot of information. And so I decided to do something a little bit different from what I'd seen people do in the literature, but I'm going to keep this in for my publication because this is kind of how people have been looking at this stuff. And so what I did is to investigate this further and to look at kind of the evidence of performance constraints and more kind of evidence of trade-offs in bandwidth and trill rate, I created a set of hierarchical models. And so I divided bandwidth and trill rate up. So I had, these were the dependent variables. And I looked to see if location of the note within a single call. So I call this bin. So if we see a difference in bandwidth from here to here, that was the first thing that I was predicting that I would see. The second one, I wanted to know if there were differences between calls that were produced during playbacks or calls that were produced spontaneously. So that's what this playback variable is here. And then also I was interested to see maybe, because I'm looking at one particular call here, but they sing over the course of maybe 15 minutes to an hour, and there are multiple calls that are happening. So maybe there's something going on over the course of a calling bout. So those were my predictors here for bandwidth. And then trill rate, I included the same things, location of the notes, and then if it was collected during playbacks or not, order of the great call. And then I had kind of an intuition, or kind of based on analysis that I did with Mark, that there might be a trade-off between the duration of the trill and the rate of the trill. So I included that as a predictor as well. And so just briefly, so here I'm showing you, these are the results. So I did what's known as model selection. And the top models for bandwidth and trill rate, I'm presenting the coefficient estimates, or the results here. And so what I found for bandwidth, so here, this is bandwidth. And so playback was not an important predictor of bandwidth. The sequence, or what the number of, the order of the call in the calling bout, was also not an important predictor of bandwidth. But we see that trill rate is an important predictor of bandwidth. So this is more evidence of this trade-off. And then also bin. So we see, and I can go back here, as we go along the course of the call, the bandwidth starts to get smaller. And then for trill rate, I found that trill duration was an important predictor of trill rate. So there seems to be a trade-off between how long they can trill and how fast they can trill. And I don't know what's going on with that. If anyone has any ideas, I'd love to talk about that after, but it's something that's interesting. And then also bin, but it has the opposite effect. And this is something that working with these calls, I had an intuition about that. It seems like the trill rate gets faster as you go along the course of the call. And so that's kind of the summary of the results of my first chapter. And so I was really just interested to see if there is evidence of this trade-off, if there is some sort of evidence of constraints in terms of what's going on with the trill rate and the bandwidth. And so just to summarize, I found that there was a trade-off between trill rate and bandwidth, but then also a trade-off between trill rate and the duration of the trill. And then, as I said, so I've found some really interesting patterns of variation, but I have more questions now. And so first of all, I'm really interested to know if we see trills that are faster, or the notes have a higher bandwidth, wider bandwidth, is this providing any information to other gibbon, either females or males? Is there any sort of information in this trade-off? And so there's been a study done where they were looking at gibbon females, and they found that there was a relationship between how long they took rest in between the notes and their age. So older females take longer breaths. And so maybe there's something going on with age, but that's not data that I have. And then I would also like to, and I think when I go back to Malaysia and Indonesia here next or in January, that I'll probably do some playbacks where I'll artificially manipulate the trill notes. And so I can make them go faster, I can make them wider bandwidth, and I can see if females respond differently. So if they're actually picking up on those differences. Okay, that was the first one. So we have 2 more. And so again, coming back to this organization here, this idea of different levels of variation in bioacoustic or acoustic data and vocalization data. For my second chapter, I focus predominantly on these 2 levels here. So how variable are calls within a single individual, or conversely, can we tell different individuals apart? And this one has been published already in the International Journal of Primatology, and is, as Meg was saying, in terms of thinking about the conservation applications, I'm not going to go too much into detail about the site where I collected the data here, but it was at this Stability of Altered Forest Ecosystem site where they're experimentally fragmenting the forest. And so my idea is to potentially use this vocal individuality to track and monitor them later. But the first thing I had to do was show if it existed or not. So I won't play these for you again. By the end, you'll be like, how many spectrograms do we have to see? But I want you to think more now about not just looking at the differences in the trills, but just with your, your naked eye as an observer. You can tell the difference between these 2, right? They look different. And I played them for you, and hopefully you could hear that they sounded different, or maybe I've just been listening to these for too long that I can really pick out the differences, but they do sound different if you listen to them for 4 years straight. So what I noticed, and what other people in the past had noticed about these gibbon calls, is that they seem like they're individually distinct. And so how do we go about determining that? Well, the first step, second step. First step is you have to go out and actually record them. And then the second step is, once you have all of your data and all of your calls extracted, you do something known as feature extraction. And so I'm showing you here, this is again a spectrogram. And so what I did with the feature extraction is that for note 1, I estimated, and it shows up here, the maximum frequency and the bandwidth. And I actually need to change this because I didn't have the minimum frequency. But I estimated some spectral or frequency parameters for each of the notes. And then I also looked at the trill duration, so the duration of this part here, the duration of the whole call. And I used 18 features to, I estimated 18 features from each of the Bornean gibbon female great calls. And so I tried to figure out about how much time it took to do that, because one of the things when I was writing my grant proposals, I was like, looking for vocal individuality, and if you can use this vocal fingerprinting to track and monitor individuals for conservation, it would be great. For 414 calls, it took me about 10 minutes per call, and that's about 69 hours of pointing and clicking. So I did it because I'm an academic. If you're a manager out in the field, maybe you couldn't do that. And so it's kind of led me down another path of being really interested in other feature extraction methods. But this one is really the standard, what we use. And also, it can give us a lot of information in terms of what's going on, in terms of the differences between individuals in note duration and these various things. And so what I found, which may be not surprising, is that there really was strong evidence of vocal individuality in Bornean gibbon female calls. And so here we have, this is a biplot of the first 2 linear discriminant functions. And so I use something that is called discriminant function analysis to try to differentiate between females. And so it's something that we call a supervised classification technique. So I told the computer, I was like, okay, here are the different female identifications, and I want you to try to maximize the difference or the separation between these females based solely on the features of the calls. And so I always, and Andy's actually here to see this, I always talk about these plots. And if you were to interpret this, and if there were no differences among individuals or females, we would see it would just look like a sneeze. There would just be points all on top of each other. But what we see is that particularly here, so each of these points represents one great call from one female, and then the circles are the 95% confidence intervals or ellipses for each individual. And so what we see is that there's one individual way over here, one individual way over here. And so there is substantial individuality, substantial variation. And then you can do something called leave one out cross-validation. So it's kind of exactly what it sounds like. You take out one of your calls, and then you run your linear discriminant function, and then you put it back in, and you try to you say, okay, identify which female this call is from. And I was very, very happy with this, actually, 95.7% accuracy in the classification. So quite good. And so this plot doesn't really give any sense about which of these features are distinguishing among or between females. And so I'm going to show you it's the same data, but just presented in a different way. And so again, we have linear discriminant function 1 and 2. And you may not be able to even see these, but each of the little points represent a female great call. And so what we see is that, and I measured temporal characteristics such as note duration, but I also measured the frequency or the spectral characteristics such as maximum frequency or bandwidth. But what we see here is that the spectral characteristics didn't really distinguish among females, but what did was variation, particularly in note 1. So how long the duration of note 1, how long or short it was, and then note 4 duration. And then also this inter-note interval, and so this is how long they take breaks in between their longer introductory notes. And again, I don't have any sense about why there might be differences between this inter-note interval. Maybe some are old and some are young, or maybe it's something else that's going on. And so coming back to this slide, so I did mention that we see there is quite a bit of separation among or between females. But then we also see in the middle here that there's quite a bit of overlap. And so I was interested to know, okay, so if we see that there are females that have calls that are more similar, what is going on? And maybe it could be that females that are closer together in terms of where their territories are, maybe they're more similar, or maybe they're more different. And so I tested, there were two kind of separate hypotheses that I tested. And so the first one is that if mothers pass on call characteristics to their daughters, and again, we don't really know anything about heritability, but if we see that mothers are passing on some aspects of the acoustic features to their daughters, and then daughters establish territories relatively close to their mothers, then we would predict to see that females that are in close geographic proximity are going to sound different. Or conversely, it may be that there is some reason that gibbon individuals would want to sound different from their neighbors. So maybe they're trying to send some information about their individual identity, and so maybe they need to discriminate or distinguish, and so maybe they would be different. And so those were my two kind of hypotheses, is that, okay, well, maybe we'll see them being more similar, or maybe we'll see them being more different. And so what we did is I calculated something that we call a pairwise distance for each call. So I took all of the features of one call and then all the features of another call, and I calculated the distance from them. And so if we have an acoustic dissimilarity measure of 0, that means that they're the exact same call. The features are all exactly the same. Whereas if we have a value that's greater than 10,000, that means that they're really, really different. So you can calculate a distance in this manner. And then I also calculated the actual geographic distance. So how far away were they from each other? And so that's what each of these points represents, a geographic distance and acoustic distance measure for a particular pair of calls. And so what we see here is, well, what I was predicting to see was something besides this line just being straight. I was expecting to see that either they would be more similar, as the females that are close together would have calls that are more similar, or maybe they were actively differentiating from each other. But what I found was that there's not really any pattern, at least at this geographic scale. And so this was at one particular site, and it was over about 8 to 10 kilometers. And so at least at this geographic scale, I didn't really see any patterns of geographic variation. But hey, non-results are still results. And so to kind of summarize here, discussion and future directions. So there is really strong evidence for vocal individuality, which this isn't something that particularly surprised me in the literature with other gibbon species. It's something that's been pretty well documented, but not in my particular species. And then, as I mentioned, I'm really interested in the conservation applications. So I have recorded all of these females in the site, and I would like to go back, and I think they have actually finished, they've completed the logging and the fragmentation, to try to go back and re-record them to see if I can identify those females. But there are a lot of complications that go along with that, which I'll talk about just in a second. And then, as I mentioned, I'm also really interested, so as I said, it was a lot of work, and Allison's done a lot of feature extraction too. That's a lot of pointing and clicking. And so I think in terms of understanding evolutionary questions, this method of feature extraction is probably the best way to go. But if you want to just do something quick and rapid, it's not. And so I'm really kind of interested in moving towards doing these more automated feature extraction methods. And then some remaining issues that need to be addressed if we're going to use vocal individuality for conservation method, or really not even just for conservation, this is kind of more big picture understanding what's going on with vocal individuality, is to know how stable is it over time. And so I recorded over multiple field or recording seasons, but I tried to go to different parts of the field site or the project. And so I don't know how stable the calls are from year to year. And so that's something that I'd really like to know. And I also, I went to a territory, and if I was trying to re-record a female, it's like, okay, I recorded her here before, so this is probably the same female, but there's a lot of uncertainty there. So there are other ways that you could identify individuals using genetic data, etc. So that's something that would be really important for these conservation applications. And then something that a reviewer brought up that I hadn't really thought about, but it makes a lot of sense is, okay, well, if we're trying to use this vocal individuality for conservation, what happens if the daughter sounds exactly like the mom, and then the mom goes and the daughter's there? We won't be able to detect that changeover. And so that's something that there's a whole lot of work that could be done, and I think very interesting work in trying to understand if these calls and these traits are heritable. Okay, and then now, third one, almost there. All right. And so for my last chapter, and this was really the chapter that got me thinking about these different levels and the sources of variation in animal vocalizations and gibbon vocalizations specifically. And again, they're not mutually exclusive, but we're going to kind of focus on the last three levels here. So looking at how variable calls are within a single individual. Are they distinguishable between individuals, which we kind of already know the answer to that. And then also, are there site level differences in call features? Spoiler, yes, there is in one, which I was really excited about. And so before I jump into this last little bit, I want to thank Mark so much for his help on this analysis. And I gave a talk at the Acoustic Communication by Animals conference in Nebraska in the summer, and people were so excited about this analysis. And so I'm really excited to get it out there and get it published. And so let me go back just to the title, and well, it kind of tells you everything you need to know. So I did what we call a multivariate analysis, and I'm going to define what multivariate means in the next slide. I've kind of already hinted at it a little bit. And then I looked at, obviously, inter-individual variation, and then in one of our features, I found that there was site-level variation. And so I don't know if a problem is the right word, but a challenge that we run into as bioacousticians, and I know it's not just limited to acoustics, but a lot of data sets have what we call a multivariate organization, or they're multivariate. And so I have here more spectrograms. That's mine. You probably maybe recognize that. But then up here, these are other different gibbon female great calls. And the only reason that I'm including this is to show that the general way that we study animal vocalizations is to do this feature extraction, and kind of which features you choose to use. It's going to be determined by your question, and also I found this pretty subjective. But what we have is that no matter which type of feature extraction method you use, you're always going to have multiple features per call. And so this is something that's a thing that we run into when we study animal vocalizations. And it presented a little bit of a challenge when we were trying to understand what was going on at these different levels. So trying to understand for all of the different features, regardless of which ones you are estimating, where is the variation? What is going on there? And so our main objective for this analysis is to learn about 3 sources of variation. And so again, I've talked about this quite a bit already, but understanding call-to-call level variation, or female-to-female level variation, or site-to-site level variation for, and we use, and I'll show you the features that we used in the next slide. So we use quite fewer features for this analysis than the others. So we just did it for 8 features, and we did it for my whole data set. So looking across all of the gibbons and all of the sites where I was able to go and collect data. And I won't spend too much time on this here, but I just want to show you briefly the features that we use. Again, all features were taken in Raven. And so we use note 1 duration, note 1 maximum frequency, note 2 duration, note 2 maximum frequency. We decided to stick with just the first 2 notes because we found that the second, or the later introductory notes, they were really highly correlated with the first 2, and so it didn't give us that much more information including them. And if we did include a lot more, Mark and I kind of had discussions about including fewer features at first. I was like, no, don't take away my features. And also the model wasn't as happy if we were including a lot. So we settled on this, but I was convinced that it's just as informative to include these 2 notes as opposed to all of the introductory notes. And then we also looked at the duration of the introduction, the duration of the trill, and then the trill rate. And so I just want to note, we measured the trill rate a little bit differently here. And so how we did that is identifying the start and the end of the trill, and then that's the trill duration, and then I counted the number of notes over that entire duration. So a little bit different than how I did in the previous analysis. And then rest duration we measured as well. Okay, and I'll go through this. I'll try to make it as quick and painless as possible. So our model. So I'm going to just show you briefly the model that we fit to our data. And we aim to explain where the variance in the features come from. So for each of these 8 features, where is the variance coming from? And so we have here, the variance in the features is equal to, we have U, which is a site level random effect, so looking at the site level differences. V here is a random vector for female, and then we have E as the call specific error term. And so that's really for the amount of model code and how long the analysis takes. I was very surprised to see that that's our model equation right there. And then we also estimated, and this is something that is pretty exciting and new as far as I know in terms of the study of acoustic data, is we estimated what are known as variance covariance matrices. And so we were able to look at these different levels, how correlated the different features were with each other. And so thinking back to this idea of trade-offs and coevolution, to understand these trade-offs and to be able to model these, it's really good. And then we use STAN, a Bayesian computational tool, to generate posterior densities of the model parameters. If you have no idea what I just said, don't worry. I'm going to explain what a posterior density is in the next slide. And so there's one important thing that in order to understand and interpret the results, you're going to need to at least understand what ICC means and how to interpret a value of ICC equals 0 compared to ICC equals 1. So ICC is intraclass correlation coefficient. And how we measure that here, so I have, again, my three different levels, call to call, female to female, site to site. And so we have the class variance for a particular feature over the total variance for that particular feature. And so if we have an ICC equals 0, that means none of the variance occurs at that level. ICC equals 1, that means that all of the variance occurs at that level. So these are kind of the important takeaways for interpreting the results. And so I'm not sure how many of you are familiar with using STAN or the Hamiltonian Markov chains, but this is a Hamiltonian Markov chain, and so we ran two of these over 500 iterations. And if you see here, so we have iteration and then we have ICC here. And so what I'm going to show you is what we call a posterior density. And so it's taking all of the values for those two chains and putting them together and creating a density plot here. And so we could have ICC way down at 0, and so that would mean that none of the variance occurs at that particular level, or we could have it up at 1. And so hopefully this will give you at least a little glimpse into what I'm going to be showing you now for the next couple of figures. And so now moving into the results and thinking about, first of all, how variable are calls within a single individual. So that first level, the call-to-call variation. And so I've highlighted the important ICCs for this level in blue. And so what I would like you to notice is that for most of the features, the contribution of call-to-call variance to total variance is quite low. And so what that means is that we have the ICCs are all over towards 0. But there is, well, there are a couple exceptions, but kind of the big exception that we see here is for rest duration. And so what this means is that from one call to another, we see that the duration of rest is highly variable. And I can go ahead and I'll show you. So just to remind you, so I added up the duration of the rest for all of these notes here. And so we were interested to see, well, maybe over the course of a calling bout, they start to take longer rests, but we didn't really see any evidence of that. But we do know that there is quite a bit of variability at the call-to-call level. And I just want to touch on really quick that we have differences here. There are some differences in terms of the note 1 and the note 2 maximum frequency. So the call-to-call variance is somewhat important, but not as important as the female to female. And then the next level, not surprising really at all, probably even for you guys, because I just told you that there was a lot of individual variation. We found that for all of our features that female to female variation was a really important source of variance. And so this was really not a super surprising result. And so I'll go ahead and I'll shift forward to the next one, which, as I said, I was quite excited about. And so now we're taking a step back, looking at the site-to-site level variance. And so what you can notice is that the red now, it's all over towards the end, or towards 0, except for this one down here. And so what that's showing is that for trill rate, the contribution of site is the most important to the contribution to the total amount of variance. And so just as a reminder to show you the different, or what trill rate is, and I won't play these for you again, but just as a reminder, I've shown you that there are differences, and now you know that there are differences among sites. And so something that is kind of interesting for me is to think about what does that mean in terms of real world application and which sites are we seeing are different here. And so you don't need to worry about the details too much here, but what this is showing the deviation from a baseline trill rate, and each of these posterior densities are for one of the sites where I went and collected data. And you'll notice that over here to the left, there are 2 of these posterior densities that are quite off to the side. And I was like, well, I wonder what's going on here. And so I went back and I was thinking about where these sites were, what could potentially be going on. And so these are the 2 sites where the trill rate is substantially different from the others. And something that for me is really interesting is that both of these are north of the Kennebetongan River. And the Kennebetongan River has been shown to be a pretty big geographic barrier for orangutans. They've gone out and looked at genetic data, and there are high levels of genetic differentiation across the sides of the river. And so my guess is that there's something going on. So the gibbons on the north part, or the north of the Kennebetongan and the south, they have been separated, and so that's why we are seeing differences in trill rate. To understand the evolutionary processes, which is something that I was hoping to kind of get into for my dissertation, that it's outside of the scope of what I can do, but I know that there are differences, and now hopefully maybe a grad student of mine one day can go out and try to figure out why. And so I think as I'm running over, I'm just going to go into the conclusions really quick. So we found substantial intra-individual variation in this duration of the rest. And again, I don't really know what's going on. If anyone has any thoughts or ideas, I'd love to talk about that. found substantial inter-individual variation. And then, of course, I already showed you my previous data about that. And then I found that trill rate varied predictably between sites. And just thinking about the future direction, so as I mentioned, I applied this analysis to one specific question, but it has broad scale applicability to the study of all sorts of different types of vocalizations. And so I really hope that people will start to kind of adopt and use this as a tool to try to understand where the sources of variance are coming in, and then maybe they can also include some ecological predictors to try to figure out as well if there are site level differences. So hopefully that will expand on our method here. And then, yeah, as I said, so we've shown that there is site level variation in trill rate. Is it due to neutral evolutionary processes and just due to the fact that gibbons have been separated on either side of the river for a long time, and we've seen this slow change into different trill rates? Or is there some sort of selection pressure in terms of what's going on in the trees and the forest to see a faster trill rate or a slower trill rate? Or is it something else entirely? So that's something that I would really like to see answered at some point. And then I'll go ahead and skip that because we are right at the end. And so in conclusion, and it was really interesting sitting down and writing my introduction, which I found was one of the harder parts of the dissertation. Like, how does this all fit together? So hopefully you guys were convinced that it does. If you weren't, let me know where I can fix it. But I do, so I summarized kind of what I thought of as being kind of the important takeaways. So I showed that there are trade-offs in the vocal production, that there are substantial inter-individual differences, and then there are also site-level differences in trill rate. And so I really just showed that there are real interesting patterns of variation. And so I hope that people will kind of work off of this to try to understand, okay, so there are these patterns, why? So either through playback studies or going out and collecting the genetic data, there's a whole line of research that's opened up for that. So, and that's pretty much what I said there. So I will go ahead and finish with that. And thank you all so much for listening.
Questions for Dena?
Yeah.
I was curious with the bandwidth trill rate trade-off. In birds, usually the hypothesis is that it either has to do with vocal tract resonance, or it's you have to open your bill wider to get a higher bandwidth, and you can't do that as fast. So what do you think the mechanism?
So this has been driving me crazy because when I was out recording them, I was hiding from them because if they saw me, they would stop singing. So I don't really have any good, video of them singing, and I tried to look on YouTube to try to find gibbons that are trilling to understand what is going on, really how they're the mechanism that's producing this trill. And so I don't know, and it's something that I've thought about what could it be, and I I would like to find out. And if you have thoughts about how I could go about that, that would be great. There is one. I actually got a grant from the Cleveland Zoo, so they do have Bornean gibbons there, and they said that I could go sometime, but of course it's the time issue. So, yeah.
This is kind of piggybacking off Gail's question. Do you think morphology would explain within species variation, and do you see morphology maybe playing a role in kind of interspecies ability to deal with drills and other?
So that's a good question, and I'm trying to think about, I personally don't really know much about the different vocal tract morphology of different gibbon species, and as I mentioned before, there are some that trill and some that don't, and they're in the same genus. And so, I mean, it would be surprising if they have really large differences in their morphology. But this is something that I realized as I was starting this. There are all these papers people publish quantifying individuality, but then they're kind of like me at the end. We're like, well, we don't know why or how this arises. But yeah, that's something that I would really like to know too, to understand. Yeah.
So piggybacking kind of when I think about the cues that I tune into when I'm thinking about identifying who's talking to me, it's often that it seems more related to the spectral characteristics of voices, although there certainly are timing ones as well. We'll say I have megapause. Anyway.
I think you just did it, actually.
So were you surprised that the spectral characteristics weren't coming across more strongly? And do you have thoughts about why that is?
I was, and I was really surprised because even in other studies, people had found that it was the spectral characteristics, so other studies of gibbons. And I remember like going and talking with Mark about this data and just like double, triple checking him, is there something, am I doing something wrong? What's going on? And I mean, to think about why we would see differences in frequency versus duration, I can kind of get back if you think about the acoustic adaptation hypothesis. You know, maybe you'll see differences in frequency based on characteristics of the forest, but then how that would influence individuals, it's kind of hard to know. And I mean, you see differences in frequency in a variety of primates and animals based on kind of their emotional state. But then again, thinking about how that applies to gibbons, and in the sense that their vocalizations are so stereotyped as well. It's like the same thing over and over and over again. So I don't know, but it really interesting things to think about. Yeah, Peter.
For the second chapter where you looked at the relationship between geographic distance and acoustic distance, I was wondering how you determined or decided on your acoustic similarity or dissimilarity metric, and is that like a standardized way of measuring distances between?
So it's something that Mark was the one who suggested I do it, but I was familiar with it before because it's kind of, I think Lydia Bedreau did it looking at ecological or environmental distance. So it's a pretty common metric that you use to look at how similar or different habitats are or vocalizations are. And so you can include all of these different features and then it'll calculate how close or similar they are. So I just kind of followed convention and Mark's advice.
I was wondering if you were happy with that distance metric, because the pattern you see could be because your distance metric is actually just giving you noise, right? And it's not actually accurately representing the distance between these calls.
Well, I think you have a really good point, but I would think it's probably, would be more related to the features that I chose, maybe than the distance measure. And this is something, I think I talked about this with Gail when I was first getting into bioacoustics. Like, it seems like it should be so objective, but it's not. And it's like, you know, which features do you choose to measure and how do you measure them from the spectrogram? Which spectrogram settings do you use? It's all of these things. So I think that there could maybe be a pattern, but I would guess it's more related to my choice of features than the choice of the distance measure. So yeah.
Is it possible to think about using methods from computer science that rather than focusing on identifying characteristics, like defining and then and measuring these characteristics, instead take the entire signal? And so I'm thinking like dynamic time warping or sort of ways of measuring distance between time series measures. This is essentially a time series measure.
And I have seen people do that same exact thing. I believe Lemurs was a paper that where they used dynamic time warping to look at differences. I don't remember if it was individuals or species. And so yeah, that's something that people are doing. And I kind of delved into it because again, this is something I'm interested in, but I ran out of time.
Does it present different problems than feature extraction? Or?
I mean, how do you interpret what's going, so it's like if you see these distances, how do you interpret what's driving them? And so as frustrated as I was with feature extraction and my arm was falling off, I think that it's the best that we have for answering certain evolutionary questions. But yeah.
Is there any data either from, for example, like your playbacks? Was your playback always the same playback?
Yes, it was. Well, except for the site where I recorded the playback, but all the other ones, yeah, it was the same. Yeah.
So then not from, like, other have a sense of what mean the recipients, right? So that's another way to be more theoretically motivated to speak out.
Yeah. It's something that people are definitely interested in. And so one that comes to mind, which I want to try to replicate it because it was done back in 1984 or 3 by John Matani, was he didn't go and play calls with kind of certain sets of features, but he went and he played calls of the same individual or a neighboring individual or an individual from another population. And he didn't see that they responded any differently. It's like they had the same behavioral response, excuse me, regardless of if he was playing the call of the focal primate or a neighboring primate. But he also said it may be due to the fact that the speakers were not that great. And so, maybe it's something that should be replicated. And I'm thinking about, I am going to be doing more playback experiments when I go back in January. And so that's something that I'm thinking about, to try to do these playbacks and figure out what information, if any, they're getting from these calls. Okay. Champagne time, 207 Cottage Circle.
Please come, we'll be there. And thank you so much, Dena.
Yeah, thank you guys.